Hello, everyone. Welcome to the ABO review section of the 2020 Blood Bank revamp. Of course, copyright here by me. Okay, so this is the blood typing section. Remember, you will always have an introductory slide that tells you what you're going to learn in this section. This section is going to cover the chemical structures of the A, B, and H antigens. The H antigen is listed first on the slide because the H antigen controls the presence of the A and B antigens, although you really don't hear about it once you learn the genetic function of it. Okay, so the main thing about the ABO genetics is that there's three genes at different loci. They influence the occurrence and location of the ABO antigens. The ABO antigens involve the ABO, the H, that's a capital H and lowercase h, and then the SE. You're going to associate SE with the secretor status. H, you're going to remember Bombay. And ABO, of course, that's our traditional blood types. A person's A, B, O, or AB. The presence or absence of the ABH antigens on the red blood cell membrane is controlled by the H gene. That's a little hidden secret that most people don't know, most people aren't aware of. This comes up as a question because it is a little secret, so you might want to remember it. So the presence or absence of the ABH antigens and secretions, that's your saliva, your tears, is influenced by the SE gene. SE stands for secretor. So if you have the SE, then you are able to have those ABH antigens in those fluids. All right, so ABO antigens are defined by what's called immunodominant sugars on the cell surface. You're going to hear that term come up, and then you're going to want to know what the sugar is. The antigens in the group exhibit autosomal codominant inheritance. That means you get A and B together. They have the same influence, so together you would have an AB. One is not more important than the next. One locus on chromosome 9 occupied by one of the three alleles. The alleles are A, B, and O. Each person has a pair of chromosomes that carry an allele. This goes back to the genetics that we've already covered, where if you have a person who gets an A from a mom and a B from a dad, that person becomes AB. If the person gets an A from a mom and an O from a dad, the person becomes AO, which the blood type will be A. Group O is considered a silent allele, which means there's nothing detectable, no product that shows up. It's the absence of A and B in essence. One thing to note is that A and B genes encode for glycosyltransferases that make up A and B antigens. The genes do not directly encode for the antigen. Genes do encode for the enzyme that transfers the immunodominant sugar that leads to the specific antigen. So this is important. They code for the enzyme, not the antigen. So glycotransferases are a group of type of sugars. Transferases means an enzyme. So it's an enzyme associated with the sugar. Transferase meaning transfer. So it transfers a particular sugar to a different part. The O gene does not code for an enzyme, so no detectable gene product is there. Okay, so A codes for an enzyme, B codes for an enzyme. The H gene, which is the capital H and a little h, has alleles. Little h is amorph. It has no detectable gene product, and that's, of course, where Bombay comes in. The Bombay phenotype lacks the capital H antigen, so it's the two lowercase h's. This is important. Bombay phenotype individuals lack both ABO and H antigen expression on their red cells, so that means they actually look like an O, but they aren't. The SC gene for secretor, and this is how you write it. Capital S, lowercase e means the person is a secretor. Lowercase s, lowercase e means they're a non-secretor. Non-secretors are an amorph. Remember, A without. So your ABO genes have their alleles, A, B, and O alleles. We mentioned that. So what is the relationship to the ABO, the H, the SE, and the LE? Okay, so the ABO, you have the alleles A, B, and O. H, you have capital H, lowercase h. Two lowercase h alleles means Bombay. SE, you have capital SE, lowercase se. The capital SE is a secretor. Remember, if you have the H, then you can express AB on your blood type. If you have the SE, you can express the ABO and H groups in your secretions. Then you have LE, which is for Lewis, which is another blood group. So the A and B antigens, this is where that sugar comes into play from a couple slides ago. The A gene codes for transferase enzyme that adds the immunodominant sugar N-acetyl-galactosamine to the terminal sugar of the H antigen. The gene product is N-acetyl-galactosamine aminotransferase, So this is now the enzyme that is produced. The B gene codes for transferase, another enzyme that adds the immunodominant sugar D-galactose to the terminal sugar of the antigen, which gives a gene product of D-galactosyltransferase. So this is the new product here. So therefore, you see the B blood type with this one. You see the A blood type with that one. I have seen this come up as a question before. Antigen concentration. Something that comes up is, how much of H is present, because H is important in telling you whether or not you actually have an ABO blood type. So most H antigens are present in the O, and then it goes down. So A2B, A2B, A1, A1B to having the fewest H antigens. So they don't have that much. The good thing is that you don't really need a lot. You just have to have them. Certain blood types possess more H than others. Group O individuals have many H antigen sites because they have no A or B antigen. Okay, let's digress and go over one thing that comes up. It's like a random question, especially if you take the technologist exam. Bombay phenotype. Discovered in India. This means the red blood cells lack the H antigen. That means the lowercase h. The H gene is amorph, results in no production of L-fructosyltransferase, which is another enzyme combined with a sugar. If you type the patient will type as a O blood type, but they don't have any H, and they don't have any A, and they don't have any B. So in their serum, they have anti-H, anti-A, and anti-B. We have something called anti-H lectin, Ulix urophaceus, which will not agglutinate Bombay cells, but will agglutinate O cells. This is how you can tell if the person is a Bombay individual. So what happens in the laboratory if we have a Bombay person? We can't give them group O. So if we transfuse them, group O red blood cells cannot be given because the H antigen is present, and that's going to cause an antibody reaction. So only thing we can use is autologous blood or donor files. So if you know the person came from India, we can actually see if there's blood from India that we can use from this little bitty island over in India. We can use that blood to transfuse the patient with. Okay, secretor status. There's two alleles, the SE, capital SE, lowercase e, SE. The capital SE is responsible for H substance and secretions. H is converted to A or B by glucosyltransferases. It's in red because it's important. This allows for the A, B, H, and Lewis, B, and body fluids. 80% of the population are secretors. This means they're either homozygous or heterozygous for the secretor status. So most of the time you're going to be able to see this expression in the saliva. 20% are non-secretors. Okay, so here's an example of your secretor. If you have a person who inherits the AB and they inherits the H, the antigen that's going to be expressed in the red blood cells is A and B and H, because the H allows for the expression of A and B. And the saliva, you're going to be able to express the A, B, and H because this person is a secretor. If the person is A and B with H and then they are a non-secretor, you're going to have the same expression in red blood cells, A, B, and H, but then you're not going to see anything in the saliva because they are a non-secretor. Take the person who is a blood type O. Remember, O does not have A and B antigens. Therefore, in the presence of H, you're going to only see the H antigen, and then you're going to see the H in the saliva if the person is a secretor. And then on the next one, you're not going to see H in the saliva if the person is a non-secretor. Okay, LE stands for Lewis. Lewis antigens are antigens which absorb onto red cells as individual matures. A person who has a H and LE but no secretor will have H and LEA, Lewis A on the red blood cells, and Lewis A only in the saliva. So secretor is not needed for Lewis A, but it is for H. However, a person with H, SC, and LE genes will have H and Lewis B. So the person has to be a secretor in order to have Lewis B on the red cells. Okay. Decreased amount of Lewis A in the body fluids. Next section. You're going to demonstrate knowledge of antigen-antibody reactions, antibodies of the ABO system, demonstrate knowledge of ABO4 in reverse typing. Okay, so let's get into ABO antibodies. What do you need to know about ABO antibodies? One, you see that it's non-red blood cell stimulated. That means they're considered naturally occurring, which means you do not have to have a transfusion in order to produce an ABO antibody. There's something in the genetics from having that H allow you to express A or B that also allows you to stimulate an antibody. Remember, you produce antibodies for what you don't have. All right, so results from exposure to A and B like antigens, they consider in the environment. So this is something that's evolving over time. So what I remember for test purposes is that it's naturally occurring, non-red blood cell stimulated. ABO antibodies may not be detected until 3 to 6 months of age. This is why we do not do reverse typing on babies. See this little bit here? We type the mom. We type the baby with forward typing, and then we go from there. Later on, 3 to 6 months of age, we can do reverse typing on the baby. That would change based on facility, and that would change based on test. Depends on what test you're taking. So remember, it's somewhere in that ballpark, 3 to 6 months of age. People possess antibodies against antigens they lack. If A or B antigen is not present, the person will make antibodies against the missing antigen. That's what I told you. Full expression of the antibody occurs between 2 and 4 years of age. I've seen some paperwork or some books or some literature that says around 5 years of age. So it just depends on what you're reading. But if you are taking a test, choose the one that's closest to 2 to 4 years of age, and you can't go wrong. So antibody titers, the titers show about how much of the antibody is there, reach their maximum levels by the time the person is 5 to 10 years of age and decrease as the individual grows older. If you understand this concept, it will help you understand why an elderly individual, I'm talking about a person in their 70s, 80s, 90s, will have not have the same expression of the antibodies, and their reverse group might not be as strong as it was when they were in their youth. Okay, most anti-A and anti-B antibodies in group A or B individuals are IgM. Why is this important? Well, because this is the time that IgM is very, 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 very important. It binds complement. Binds complement means that it can cause a transfusion reaction immediately. So it reacts at room temperature, immediate spin. So when you first put the patient's blood together, if you give this unit of blood to the person, in a few minutes, if it's an ABO incompatibility, you can have a transfusion reaction. Anti-A, B antibody is found in group O individuals. It cross-reacts with both A and B antigens. And here's what you have on the chart. If a person types as A, that's this one right here, they have an anti-B antibody. It's going to be IgM, a little bit of IgG, but mainly IgM. If a person types as group B, they're going to have an anti-A antibody, which is IgM, some IgG. A person who types as group O, they're going to have 3 antibodies, anti-A, anti-B, anti-A, comma B, mainly IgG. Okay, so what this says is that if my blood type is A and you give me B blood, I am definitely going to have a reaction. Okay, routine blood typing, phenotyping. So when you do phenotyping in the laboratory, there's 2 tests that are done. The tests are the forward grouping and the reverse grouping. The forward grouping, red blood cells are tested for ABO antigens. Remember, the antigens are where? Red blood cells have antigens. Determines the ABO phenotype. Reverse grouping, serum or plasma, is tested for ABO antibodies. Acts as a control for red blood cell testing because these 2 should match. In essence, they would be the reverse of each other. A key is that both forward and reverse testing are required on donor and recipient blood samples. You have to do both. And this is according to the standards for blood banking and transfusion services. So reagent anti-A and anti-B testing is performed at room temperature. This becomes important because, of course, you can ruin the antibodies if you are performing it at the wrong temperature. A little bit more about the forward typing. Unknown cells are added to known antiserum. The unknown cells come from the patient. Okay, so this is the patient. The reagent antibody is found in the antiserum. We know what the reagent antibody is. If there is no agglutination, it means that the cells lack the antigen to which antiserum corresponds. If there's agglutination, it means the cells possess the antigen to which antiserum corresponds. Okay, because antigens are found on red blood cells. That's the forward typing. And it is important for you to know these terminologies, forward typing versus reverse typing. Here, reverse typing. Patient serum added to saline, suspended known group A1 and B cells. No agglutination means the patient serum lacks the antibody to the antigen on the known red blood cells. Agglutination means the patient serum possesses antibody to the antigen on the red blood cells. A side note, best reactivity occurs at 4 degrees Celsius. So if you have questionable reactions, you can incubate the tubes at 4 degrees Celsius to increase reactivity. So if you do have a child, maybe you see that 2 to 4 age range, and maybe at the place you're working, they tell you that when they're 18 months, you can perform reverse typing. You perform reverse typing and you don't get reactions. Well, if you incubate it at 4 degrees Celsius, the reaction should become stronger. In the case of when you have reactions that are not showing up in a reverse group, you can do what's called a mini code panel. So I gave you guys a list of ABO discrepancies. It should be attached to this module that you have. You can open it up and you'll be able to refer to that one when it comes to this section. Why do you do a mini code panel? To enhance the serum anti-A and anti-B reactions when they are expected but are not demonstrating at room temperature. It's used to help identify cold antibodies reacting with other antigens on A1 and B reagent cells. And if the person is older and they have a weak expression of the antibodies, it will actually improve your reactions. If you're taking a technologist exam, you might want to know what's involved in a mini code panel. A mini code panel means you test the patient's serum with A, B cells, O cells, O cord cells if they're available, auto control. Based on your results, you have a certain interpretation. If you test and you get positive in the A and B cells, positive in the O cells, negative with the cord blood, positive in the auto control, the patient is considered to have an anti-I. Anti-I is something, it's an antibody that occurs, and it's a cold antibody. It's a common cold antibody. So it's good for you to know that from a historical standpoint. A person can be positive with AB cells, positive with O cells, positive with the cord cells. So now the difference is they're positive here. Negative with the auto control. It means they have an unexpected antibody reacting at cold temperature. It is considered an allo antibody because the auto control is negative. This I, it would be considered auto anti-I because the auto control is positive. Here, if you have AB cells positive, negative O cells, negative O cord cells, negative with auto control, that's either anti-A or anti-B, which is what you want to see if you suspect that the person does not have strong antigens. Okay, ABO phenotype reactions. All right, so phenotype group A, that person has in the forward type anti-A, anti-B, that person gives a positive with the anti-A. In the reverse type, the person gives a positive with the B cells. Remember, it's always going to be the opposite of what you had initially. So this is the forward type. This one is the reverse type. Group A, positive with A. Group B, positive with B. Group O, negative. And AB, positive with both on the forward type. And then the reverse is the opposite. Another thing that we will learn, demonstrate knowledge of subgroup typing for A. Subgroups of the A type. We have A1 phenotype, A2 phenotype, you have an A3, you have an AX. There's a lot of different ones. The main subgroups of A are A1 and A2. Both react strongly with reagents, and A, they give a 3 plus to 4 plus reaction. To distinguish A1 from A2, we have a lectin called dolicholas biflorus. It's used, it's an anti-A1. It agglutinates with A1 cells, does not agglutinate A2 cells. 80% of A or AB individuals are subgroup A1. That's the main group. That's why it's number 1. 20% are subgroup A2. That means they will have an anti-A1. On the A1 phenotype, here's a little thing here. Positive with anti-A. Positive with anti-A1 lectin. The A2 phenotype, positive with anti-A, negative with anti-A1 lectin. Why is it important to know about subgroups? If you have a weak subgroup, it's missing in a donor, the incorrect blood group could be given to a patient, so transfusion reaction. So a group A subgroup in the donor is classified as group O. The group O recipient gets a unit of group A blood, has a transfusion reaction. So because they have anti-A antibody. Anti-A1 antibodies produced by the subgroups of the A type has specificity to the A1 antigen but will not agglutinate A2 cells. Not clinically significant, but it can cause incompatible cross matches on immediate span testing. Reactivity with anti-A1, a A1 cell will be positive with anti-A1, a A2 cell will be negative with anti-A1, a A3 cell will be negative with anti-A1. So in essence, we say a person is A1 if they are positive with the anti-A1. Other subgroups of A and B types, they're rare, but they include A intermediate, that's AINT, A3, AX, AM, AN, AL, band 2. You just need to know those from a standpoint of there are other subgroups. Subgroups of B are less common, and they typically do not react strongly with anti-B reagents. A rare subgroup of A may be present if you have weak or no agglutination with commercial anti-A and anti-A, B. Anti-A1 is present. Anti-H causes strong agglutination because anti-H antibody does occur as a cold. So once you know the patient's blood type, the next thing that you have to consider is selecting blood for the patient and what makes blood compatible. In a red blood cell transfusion, the ABO identical unit is preferred. So when we do the forward and reverse typing, we determine if the person's blood type is A, B, AB, or O. We like to give the person the same blood type. If they're A, we give them A. If they're B, we give them B, and so forth. There are times when we don't do that, and we'll have to learn when those times are, when it's appropriate. Incompatible red blood cells can cause acute hemolytic transfusion reaction. We're going to get into this more in later sessions, but this will become important. There's always a question about transfusion reactions on the exam. ABO identical unit is required in whole blood transfusions. We do not use whole blood anymore, but it is a common thing for people to kind of get confused. A whole blood unit contains the blood, the red blood cells, and the plasma. So that's the portion that has the serum, the reverse grouping, and the forward grouping. So the only thing that you really can give is blood that matches without actually causing an issue for the patient. Group O is considered a universal donor. So we're giving red blood cells. So Group O can give to a patient because there's no plasma in the packed red blood cells that we give, that we've taken that off. But Group O doesn't have the antibodies that we find in either A or B or A, B. Group AB is considered a universal recipient because they have A and B antigens. They can receive anything. They can receive A blood, B blood, or O blood. And plasma transfusion, remember plasma is the reverse. That's the reverse typing. That's what we're dealing with. So compatibility is based on what the reverse typing looks like. ABO identical unit is preferred with the reverse red blood cell transfusion. Donor unit antibodies have to be compatible. And in both cases, you have to find out if a person has an antibody and make sure the antibodies are compatible. Group A for plasma is the opposite of red blood cell transfusion. So if you think of it, you can think red blood cell transfusion, go by the forward type. And plasma, you go by the reverse type. Okay, so the plasma transfusion, donor unit antibodies must be compatible with recipient's red blood cells. Group AB being a universal donor, Group O being a universal recipient. So on plasma, fresh frozen plasma, Group O can get any type. So what does ABO compatibility look like? So recipient, that's the patient. The phenotype, if the recipient's A, whole blood, they can get Group A. If it's red blood cells, that means the plasma has been removed from the whole blood. They can get A or O. If it's plasma, fresh frozen plasmas, they can get A or AB. Group B, whole blood, they can only get B. See, notice the whole blood, it matches. Whatever their type is, that's what they can get. Red blood cells, of course, whatever their type is, they can get that. But everybody can get O. And then when you get to AB, they can get any of them. Plasma, notice anybody can get AB, and then they get their type. And then O can get everything. ABO discrepancies occur when the forward grouping does not agree with the reverse grouping. Discrepancy may be present if Agglutination is weaker than expected. That's where I showed you guys that mini panel. Expected reactions are missing, mini panel. Ester reactions are noted. So if you have something there that's not supposed to be there. Now, one of the things I want to make sure you remember is that when you're doing the forward typing and the reverse typing, your reaction should be either 3+ or 4+. When you get a reaction that is not 3+ or 4+, say it is 1+. That's normally the questionable reaction. So if you're looking at taking a test and you see something that doesn't look right, forward doesn't match the reverse, look at whatever gives you the odd reaction, the weakest reaction. That's the one that's probably causing the issue, and that should help you identify what the problem is. So guidelines for investigation of ABO technical errors. One, let's look for documentation errors. Do you have the correct sample identified on all tubes? Did you record the results properly? So are the results in the right space? Now you're probably thinking, well, why do I need to know this if this is something that it seems like I would be doing in the actual blood bank? Well, when you take your exam, there's going to be a question, a quality type question. And the question might have something like this. And it normally is only one question. But you need to be able to answer that one question because every question counts. I do know of a person who failed their exam by one question. Okay, so you need to know this even though it seems trivial. Okay, interpretations are not accurate or properly recorded is a problem. Reagent or equipment errors can be a problem. If QC wasn't done, if the reagents were contaminated or hemolyzed, if the centrifugation time and calibration are not confirmed. Standard operating procedure errors. Procedure follows manufacturer's directives, but the person did not. Okay, correct reagents were used and added to testing. If that doesn't happen, you can have errors. Red blood cell suspensions are at the correct concentration, 2 to 5%. Cell buttons are completely suspended before grading the reaction. So these are, if you don't do these things, you can cause some discrepancies in your blood type. All right, so problems with red cell testing. Extra antigens. Group A with acquired B antigen. BA phenotype. Polyagglutination. Rouleau. Okay, so these are some common reasons for you to have problems with your red blood cell testing. That is the forward group. Hematopoietic progenitor cell transplants. If a person has received a transplant, red blood cells, even other organs, you're going to have problems. And you should know your patient's history before you get started in investigating any of this. All right, missing or weak antigens, what does that look like? Normally a blood subgroup, ABO subgroup, or a pathology or a transplant. So pathology being certain diseases that a patient has will cause them to have missing or weak antigens, and then also certain drugs that the patient takes. Mixed field reactions can occur. Mixed field, where you see two different populations of cells, transfusion of group O to group A, B, or AB, mass transfusion, hematopoietic progenitor stem cell transplant. So if a person has a stem cell transplant, A3 phenotype. A3 phenotype is classic for mixed field. Problems with serum, that's the reverse grouping. Extra antibodies. This is normally what we see in the blood bank. A subgroups with anti-A1. And then we have to test with the A1 lectin. Cold alloantibodies, cold autoantibodies. That's where that mini cold panel comes into play. Rouleau, we have something called saline replacement where we can get rid of rouleau. Intravenous immunoglobulin, IVIG, so a person has had a shot of immunoglobulin. Missing or weak antibodies, newborn, elderly, those are the two most common that we see, and probably on an exam, that would be the answer. Pathological etiology and immunosuppressive therapy for transplantation, so drugs. So here's a couple of examples. Remember, I gave you a handout that is in the module that you need to go through. It has more details about discrepancies and blood typing and subgroups and how to figure out what is what. So extra antigens. Here's group A with acquired B. So the group A immunodominant sugar is altered by a bacterial deacetylating enzyme, which means it removes the acetyl group from the enzyme. It looks like group B and cross reacts with anti-B. What does that look like? You have the forward type, patient red cells with anti-A 4+, anti-B 1+. So this is the forward type. I'm just going to put the F here. This is the reverse type. Patient serum with reagent red blood cells. Remember what I told you? Look at for what gives you a reaction that's not normal. Here, the 1+ reaction is not normal. 4+, that's normal. So anti-A, if it's the forward is anti-A positive, we expect for the reverse, the opposite, to be true. So over here, change colors here. 4+, 4+. That's normal. That's what we expect a person who types as A to look like. The thing that is not normal is this 1+ reaction. So this is considered an extra antigen, and it is reacting with anti-B. So normally we would automatically think that this must be a group A individual with an acquired B. That's what it's called, and you would see that as your choice on your exam. So why does a person have a group A and an acquired B? Why do they get that? Well, the acquired B antigen is a group A individual possessing extra antigen in the red cell testing. Weaker agglutination, you saw that anti-B is observed in the serum. Serum testing reactions are typical for group A individual. How do you resolve the ABO discrepancy? Find out the patient's diagnosis. That's always going to be your first step. Test the patient's serum against autologous red cells. Anti-B in the patient's serum does not glutinate. Here's the key. Autologous red cells with the acquired B antigen. Test the red cells with additional monoclonal anti-B reagents. So you get a different manufacturer and you test with their reagents, and then document it not to react to with acquired B antigen. Patients should receive units of group A red cells for transfusion. So you're still going to give them A blood. Okay. Next extra antigen is B or A phenotype. So it looks like acquired B, however, the patient is a group B instead of a group A. So here's an example. We're going to go back to, here's the forward type, here's the reverse type. In this case, the strongest reaction was 4+ with the anti-B, and on the reverse, 4+ with the A1 here. So does this look right? Yes. Then on the other side, we have anti-A, and we get this reaction here, 1+. That one is what is sticking out as possibly not being correct. All right, so what happened? The agglutination of the red cells with anti-A is weak. The agglutination of the patient's red cells with B is strong. Results are typical of a group B individual. And the B phenotype of group B with an apparent extra antigen is observed with the anti-A. How do you resolve it? Look for the history. Test red cells with additional monoclonal antibody, anti-A reagents from other manufacturers. All right, other reasons you can have extra antigens: polyagglutination. Poly means multiple agglutination. Hidden antigen on the red blood cells is exposed and reacts with most human serum. Normally you don't have this happen, but it could happen. Non-specific aggregation, rouleau, abnormal amounts of serum, what's called Wharton's jelly. And just from a history, you need to know this, especially if you're going to take the technologist exam. So it's a contaminant that's found on cord blood cells, so you can get a false positive. So normally those baby samples that you receive, you have a false positive there. It used to be a common question. It's not seen as much, but it depends on what test you get, you might find it. Now that we just talked about extra antigens, the other side of the coin is missing or weak antigens. So some subgroups demonstrate weak or no reactivity with the anti-A or anti-B reagents that's in your reverse grouping. Patients with leukemia, Hodgkin's disease show weak antigens. Lack of glutination is an obvious issue. Patient appears to be O in the case that's above. So here we go with our forward typing and our reverse typing. So in the first part of it, these are patient red blood cells, negative, negative. And then on the reverse, we have a 3+ here, and then we have a negative here. So if this is 3+, we would definitely expect for it to kind of match up, and with the forward group, I think the 3+ is real. So there's no agglutination going on. So serum testing looks like a group A. The forward looks like a group O. So what happens? Missing or weak antigens they suggest a group of A or B. Subgroups of A occur in less than 1% of a group A population. Subgroups of B are rare. So if it's a subgroup, it's probably going to be a group A subgroup. So always check diagnosis. Repeat with an extended incubation and include polyclonal anti-B or monoclonal anti-B. And the extended incubation should cause that antigen to show up. That mini code panel helps too. Okay, so let's say in that case we just had, if we took patient red blood cells and we typed it with anti-A, B, and we actually got a reaction. So this is probably a subgroup of A. Okay. Patient red blood cells, we have anti-A, B. If we get negative, it's probably not the subgroup, so we need to do an absorption or an elution with anti-A. So that's because there's something else going on with antigens, antibodies. You have to do more work. Mixed field reactions. Mixed field contain agglutinated and unagglutinated cells. And the best way that I can describe that to you is that here's your tube. In your tube, you're going to have a population of clumps. And then you're going to have everything else. I'm going to change the color. Now you know it's not blue, but everything else is going to be clear. And then I'm going to make it, just for the sake of showing a different color, you're going to have another set of clumps and another set of clumps. So it looks like two different populations of cells here. So that's why it's called mixed field. There's agglutinated and unagglutinated cells, clumps and no clumps. So why do we get those? One, normally, if you give a person who's one blood type a different blood type. So if you give group O to a person who's A, B, or AB. The next one, which is the more common one, is a bone marrow transplant or a stem cell transplant. A3 phenotype, and this is key if you have a question about a subgroup and it tells you that it was mixed field reactions, most of the time it's A3. And then polyglutamine red blood cells mutations and hematopoietic tissue. Now, the first thing that happens is you normally see in the hospital setting, a person had massive transfusion, And then this is what happens. If a person has not had massive transfusion, next thing you see is the person maybe it's a bone marrow transplant, stem cell recipient, or one of the phenotypes. Another thing with extra antibodies is extra antibodies occur and they cause problems in reverse testing. That's where we have that anti-A1. The other thing can be cold alloantibodies. That anti-I is a cold autoantibody. And then Rouleau, which gives a false positive agglutination. Cold means it reacts at room temperature or below, normally 4 degrees Celsius. It shows up stronger. So Rouleau. Rouleau is stacks of coins. So when you see your cells under this microscope, the coins will be stacked, the cells will be stacked like this. And they call that stacks of coins appearance. So that's Rouleau. So if Rouleau is present, if we incubate the test serum and red blood cells, centrifuge for a minute, remove the serum, and then we take and mix the serum with an equal volume of saline. So however much serum we took off, we replace it with saline. Centrifuge it, spin it down. If you have no agglutination, that means the problem is due to Rouleau. If you have agglutination, it means it's real agglutination and you can trust the reaction. This is called saline replacement. You're replacing the serum with saline. Okay, so your stacks of coins disappear when replaced with saline. Rouleau is associated with multiple myeloma and Waldenstrom's macroglobulinemia. That will come up again in hematology and in serology. Missing or weak antibodies. A little bit more information. They show weak or negative agglutination in the reverse phase of testing. Reactions may be explained by investigating the patient history, age, diagnosis, and immunoglobulin levels. Newborns, elderly have reduced antibodies, pathological states, lower immunoglobulin levels. So remember, you have that entire discrepancy worksheet. Here's another one. Acquired B, this is what the reactions look like in the forward typing and the reverse typing, and this is how you resolve it. Acquired B, 4 plus, questionable reaction here. Test with autologous red blood cells, it should get a negative reaction. BA phenotype, here's the questionable result here. Test with monoclonal anti-A from other manufacturers. Subgroup of A, notice everything is negative. This is the one that we actually trust, the 3 plus here. Do extended incubation and repeat. Group B transfused with group O, so you get mixed field reaction. Check transfusion history. Group A2 with anti-A1. So A2, there you go, 4 plus. Here's the questionable reaction. Test with anti-A1. Cold autoantibody or antibody, 4 plus, 4 plus, 1 plus. Perform antibody screen with auto control or mini code panel to see what's underneath. Rouleau, saline replacement. Everything's agglutinating, but you see these 2 plus reactions are the questionable ones. Weak or missing antibodies, check the patient age. You have nothing here. Something should happen. Incubated room temperature for 15 minutes or 5 degrees Celsius and see if you get any stronger reactions.